So yeah, thanks a lot for the kind introduction and also for the invitation to talk here. HiTSeq really is one of my favorite conferences, and so I'm very happy to present my work here. Yeah, I usually start with an introduction to RNA. Now for today, I don't think that's necessary. You have heard quite a bit of RNA already. So I want to start by introducing the team who actually did the work, and I will be talking about work from PhD students. That's Christopher Hanja, UK1, and also Minhao Ling. And also work from two postdocs, that's Andrew Sim and Chen Ying. And Chen Ying also has a poster here. She's actually at the conference as well. We're located in Singapore, so this is one of the few cold and rainy days that we have. There's not too many of those there. That's because Singapore is pretty close to the equator. In fact, so close that the area where the Genome Institute is located is called 1 North. And that's because it's 1 degree north of the equator. And yeah, I should mention we have postdoc opportunities also for PI positions. If you're interested, feel free to talk to me. Okay, so for those who haven't been to Singapore, and this is where it's located, it's quite far away. And this is how it looks. It's a very nice place. If you ask some of my team members, they'll tell you Singapore is a lot about the food. There's a lot of discussion what kind of food and what's the best food. But it's a very typical scene in Singapore is this year. This is what we call a hawker center, and that's where people meet for lunch and for dinner. And that's how it usually looks, except for when it didn't look like that. And I think we all remember those days from the COVID-19 pandemic. So Singapore usually is very busy. We're not used to seeing it so empty. Luckily, the pandemic ended relatively quickly, and Singapore and all major economies really could open up again. And so by 2023, we could see really same visitor numbers as before the pandemic. And so one of the reasons that the pandemic could be ended so quickly is mRNA vaccines. You can see the numbers here. These mRNA vaccines, they reduce the risk for infection, for hospitalization, and also for death. On the right side, that's the vaccination numbers in Singapore. And it's really the high vaccination rate that helped opening up Singapore and also other major economies. And so at this point, I still want to go back to introducing RNA a bit. And so RNA is really the molecule that interprets the information that's stored in the genome. And it then translates that into a function, and that function could be a function RNA, or it could be a protein. And so RNA viruses use that as well. And so the idea of an RNA vaccine is relatively simple. One wants to use some part of the RNA to generate an immune response so that antibodies can be built. Even though the idea is simple, the problem is it didn't really work for a long time. And so I want to go back to an experiment from Katalin Karikó and Drew Weissman. So what they try to do is they try to transfect RNA into cells to generate a protein. But when they did that, they saw that there is a very strong inflammatory response, but the protein was not produced. And so if one would do that experiment with RNA that's transcribed from cells, we wouldn't see that. And so there's a difference between RNAs that are transcribed using in vitro transcription or from cells, even though the sequence is the same. And that, of course, leads to RNA modifications. And so what they did is they replaced these unmodified bases with modified bases. And that essentially solved the problem. Now they could transfect RNAs into cells, and they reduced the inflammatory response and increased protein production. And that's really the foundation of RNA vaccines. And so they're very successful. They're estimated to have saved tens of millions of lives. On all currently approved mRNA vaccines use these base-modified RNAs. And for their work, Katalin Karikó and Drew Weissman received the Nobel Prize. So what are RNA modifications? This shows 3 of them here. That's M6A, M1A, and pseudouridine, but there's really many more. There's more than 170 that have been described. They're regulated by writer, eraser, and reader enzymes that write, remove, or that interpret these RNA modifications. They can be dynamically regulated, and they're involved in a huge number of different functions. Just to illustrate some of those, this here is a tRNA molecule, and it illustrates some of these modifications that are known. You can really see how RNA is a highly modified molecule. RNA modifications also occur at mRNAs. That's shown here with the different RNA modifications that we know where they occur and how frequent they are. But one can also use unnatural modifications. For example, one can use them for RNA structure probing or metabolic labeling. And of course, RNA modifications are used for commercial RNA products, such as the RNA vaccines that I've introduced. I want to talk a bit more about one modification, that's M6A. That's because we know quite a bit about the M6A modification. The first is we know there's a certain preference. M6A occurs in this motif, which we call DRACH, or it's the GGACT chimer. There's a preference towards the 3' end of the coding sequence. And for M6A, we have good technologies to profile it transcriptome-wide, as shown here for the MYC gene. And so these figures are actually taken from a paper that shows that one can use a small molecule inhibitor to target the writer of M6A, that's metal 3, and you can see that that leads to a reduction in M6A. And the reason I like that paper is because it really illustrates both what we know about M6A, about the technologies, but also it shows an impact of targeting those modifications. In that case, it leads to a reduction in cell growth for acute myeloid leukemia, really highlighting also the impact that these modifications can have. So how are these modifications profiled? In one way or another, the idea is that one extracts RNAs and then uses some kind of trick to identify what positions are modified, which is then coupled with sequencing. Now, those often are specialized protocols. They can be expensive or time-consuming, or sometimes not scalable. And so for a computational lab like me, that means that these are actually very difficult to access. Now, even if I could find a collaborator, we can only profile very few modifications, and that has really been a bottleneck. And one of the big breakthroughs, of course, is direct RNA sequencing. So the technology works by putting a nucleic acid through this nanopore, and at each time point, we can measure an electronic current signal, and from that signal, we can identify the sequence that goes to that pore. But because we can sequence RNA directly, we can also get signal deviations from modifications that pass through that pore. And from those, we can then identify modified bases. So that means direct RNA sequencing really is a scalable assay that we can use to identify modifications. And that also means that identifying these modifications is largely a computational task. And so for those who are not familiar, I want to start by introducing a method that we have developed that's called M6ANet, which I really think is a very nice introduction to that topic. It looks a bit different from the plot before, and that's we don't know which of these RNA molecules, which of these reads are modified. Usually, we have information at the site level, which means we know at this position some of these RNAs are modified. But the data that we have is for individual RNAs, for individual molecules. And so there's this mismatch that we usually don't observe. Means we have label data for the site level, but the data that we have is for individual RNA reads. Now that's known as a multiple instance learning problem. And so I'd like to explain that with this example here. Imagine there's 3 security guards, and each one of them has a keychain, and 2 of those security guards can open the building. But the reason they can open the building is because of individual keys. Now we don't know which keys those are. And so in that case, the keychain, that's what we call the bag. The individual keys, that's what we call the instances. And that's the same that we have for RNA as well. Here we call the set of RNAs that are transcribed, that will be the bag, and each RNA, that's one instance. And only some of those RNAs are modified. And so that framework is what we implemented in M6ANet. So very briefly, it uses the signal data and also the sequence data, which is then mapped to a high-dimensional space using this read-level feature encoder. We then map that to a probability for each read, for each position that there is a modification, which is then summarized to the site-level probability. Now, the beautiful part is that we can do this only using site-level labels, but we can still infer for each read, for each position, the probability that there is a modification. And so to illustrate that here for the MYC gene, this is one of the protocols that we can use, for example, for training these models. And this is the predictions that we get from direct RNA sequence data, and you can see that it's really similar. So this is just one example. We, of course, do that for evaluation with a cross-validation as well. You can see here with M6ANet in blue, or on the right side in pink, that the performance is really good. And that works well on other data sets as well. So there's very high generalizability for these models. And that's what we see. So we can train these models with site-level data. We can predict site-level modifications, also single read level probabilities, and that generalizes very well. So one of the big advances are these base calling models, in particular the one from Oxford Nanopore that you might have heard of, that's Dorado. Now, Dorado also includes modification models, M6A, M5C, pseudouridine, and also inosine. And the advantage, of course, is that this might be much easier. You might get that directly from the sequencing run. There's still a few limitations. For example, not all modifications might be in there. And it includes the M1 pseudouridine, for example, that's used for the vaccines, or any of those unnatural modifications that one might want to use for certain genomic assays as well. And then the training data for these base callers can be more costly. It's here usually based on chemically synthesized random oligonucleotides with specific modifications. Now, those are costly to generate. And so one of the questions we had was, can we train modification models for other modifications using an approach that's more cost efficient? Now, M6A-NET was designed for M6A, but the framework is really generalizable to other modifications as well. And so that's what we wanted to use here. And what we did is we generated IVT data, and other people have shown before that this can be used. And so we use here a library that represents all k-mers. We sequenced that with unmodified bases and also with base modifications for 9 different modifications, representing all of the 4 bases. So we did then direct RNA sequencing, and we can see here the unmodified data generates the highest throughput. Once we insert 100% modification, that impacts the throughput, but we get sufficient data to train these models. And one other aspect is because we have all these modifications, we can actually learn a model that can possibly discriminate different bases at the same position. So we call that ModNet. It's an extension of M6A-NET towards a multi-class classifier. In principle, it works by using data with labels, and we can use data for multiple modifications so that we can learn this multi-class classifier. And so one of the advantages is that in principle, we can test if that works with data that's very cost efficient to generate, such as IVT data. And so some of the questions that I want to ask here is, how accurate can we use that to detect other modifications? Can we discriminate multiple modifications? And then also, what is the trade-off when we use IVT data for model training? I've introduced Dorado, and Dorado works very well. So I want to show that here for M6A. This is on IVT data, and you can see Dorado really very well identifies all those M6A positions. So this is the model that we train on IVT data. IVT data, of course, is more limited, but you can see that overall the performance is still very good. Now, we don't try to train a model on IVT data to detect M6A, but really what we can do now is we can use this framework to learn models that can identify other modifications, which are not represented in some of these frameworks, such as Dorado. And so this is how it looks. And you can see we can really detect quite well all the modifications for which we generated data. And so overall, we can see that direct RNA sequencing, in that case, the ModNet framework really can identify all the modifications for which we generated data. So now let's look into this case of discriminating 2 different base modifications. Before I always talked about unmodified and modified, but can we detect different kind of modifications with direct RNA sequencing data? And so can evaluate that using this framework that has seen all these modifications for training. And so this is how it looks for the A modifications. The first row here, that's the modifications against unmodified and a pairwise comparison. You can see that performance is very good. Now here you can see that's when you compare M6A and M1A, and that performance is much lower. So it seems that this is actually difficult to discriminate. Now, how does that look for the other modifications? Well, it turns out for C, this actually works very well. Now, if we look into the U modifications, for which we have different modifications, it still works very well. We can actually very well classify those different modifications. So the question is, what happened here with M6A? And this has been reported before that M1A can be converted to M6A during in vitro transcription, and we validated that as well with mass spec. We can see that that's exactly what happens. We have a bit of M6A in the M1A sample, and that means that's really a bit of a noisy training data set, and that explains why it's difficult to discriminate those 2 bases. Now, this is a bit of a theoretical idea. There's maybe not too many positions where different modifications can occur, but it has a practical implication as well. What happens if we try to infer M6A and there's an M1A position? Those might be identified as false positives. And so that's what we can see here. In orange, that's Dorado. This is a precision recall plot when we want to identify M6A against A, but there's also M1A now, which we don't consider an M6A modification. And the M6A model in green, that's the single modification model that we train. And if we expose those models to different modifications, you can see that really increases the precision. For M5C, it's the same. We can really increase the precision. You can see that Dorado identifies a few false positives based on other modifications, and most dramatically for the pseudouridine modification. All the other U modifications are identified as pseudouridine, even though they are not. And so you can really see how the presence of alternative base modifications have a very strong impact on these single modification models. And so training these multi-modification models that are exposed to different modifications really increases the precision here. So one question is, how does that look if we apply that to cellular RNA? And again, I want to start with this plot showing Dorado, which we know has a high accuracy. We also have M6A-Net, which is similar to the ModNet framework, only was trained on human data. And we can see here the precision of M6A-Net is actually very good, particularly here. And then Dorado tends to be more sensitive. Now, how does that look if you use the model that was trained with IVT data? And you can see there's a bit of a drop in performance. Now, the question is this good enough so that we can use those models actually? And it's difficult to say, but one thing that's clear is that these models are trained on data that's very limited. And one thing is that there are certain k-mers in the genome that they were not exposed to. In particular, all those k-mers that consist of a modified and unmodified base. And so we can split that, and that's exactly what we can see here. Any k-mer that consists both of an A and an M6A, these models have never seen them in the IVT data, and that's where the performance is very low. Now, if we look into those k-mers that these models have seen that only have a single A modification, the model trained on IVT actually works very well. And so that's where these models can actually be used. We can even quantify the stoichiometry using these IVT models. And so we do see that that works. It doesn't work as well compared to having better training data. But on the other hand, of course, this is a much more cost-efficient way. It allows us to train models for a lot of different modifications. Just to show how this looks, if you apply this M6A model to human data, we can identify this typical enrichment at the 3' end that I've shown before. If we apply the M1A model, which you know is a noisy one, we can actually already see a difference in enrichment here at the 5' end, which is a bit of what we expect. And so it shows really that we can do potentially a lot of interesting things with these models, but it also highlights really that we have to be careful with interpreting them as there might be a large number of false positives still with those models. Okay, so to summarize this part, I've introduced ModNet, which extends this MRL framework to multi-class classifiers, so we can discriminate and identify different RNA modifications. We do see that better data, of course, gives better models, but IVT data provides a cost-efficient way to train base modification models. And a bit of a look forward to what's important, really training and evaluation data is one of the key things in order to understand what we can do with direct RNA sequencing. That includes approaches that people already use, such as synthetic RNAs or modified sites from cellular RNA, whole transcriptome IVT, and unmodified samples, or these detailed maps with modifications. And all of that will be really helpful to really understand what we can do with these modification models. Okay, so I want to go back to M6A briefly for the next part, and that's because we know for M6A these two writer enzymes. M6A was first described in the 70s, and then in 1997, metal-3 was described as a writer enzyme, and in 2014, metal-14 was identified. And so the question is, this is 40 years of research. How could we do that with current technologies? And could we do this more systematically, for example, using a screening approach? Now, one could potentially use direct RNA sequencing data, but if we use these models that I've introduced, these supervised models that detect M6A, that means we already have to know a lot about M6A. So we cannot identify M6A writers in an unbiased way. However, there's an alternative approach as well, and I want to briefly introduce that. This is a method we've published a while back. It's called XPOR, and it can also be used to identify RNA modifications. It works by looking into all those reads from different samples, and they'll follow a Gaussian distribution if they're unmodified. And if they're modified, we expect a bit of a shift. And so that corresponds to Gaussian mixture model. Now we know a bit about the unmodified distribution, and that's what we can use as a prior. Then we can estimate the parameters for this Gaussian mixture model. We can then go back to those reads and assign them to each distribution to essentially identify the fraction of modified reads for each sample. And we can do that without any knowledge about any of these RNA modifications. And so what we wanted then to do was doing a direct RNA sequencing-based screen where we target RNA binding factors to identify what are possible modifying enzymes. So we selected these candidates here. Now with direct RNA sequencing, we need a lot of RNA. So we designed 2 different guide RNAs for each one of them, but we decided to pool them so that we get sufficient sequencing depth and enough RNA for each of those. We submitted that for sequencing and then analyzed that using XPOR. So we looked into the knockout efficiency, and we do see that we target the right candidates. And when we look into the sequencing data, we get good throughput between 6 to 9 million reads, very high mapping rate, and also a good read length. And so we proceed with this analysis using XPOR. Now, if we run XPOR, that's unbiased, which means we can analyze each base in the transcriptome, and that gives us about 7 million sites that we can test within each sample. And that's a huge number of tests to perform, and that limits also the sensitivity. So we decided to summarize this here by Kamer, the assumption being that an RNA-modifying enzyme would target a specific base. And then within each sample, we normalize it to essentially try to identify outlier Kamers. And so that's what we then use for ranking, and this is the plot that we get from this experiment. And you can already see there's a bit of variation here. There are some Kamers that are outliers, but really there's 4 of them standing out. So you can look into the Kamers and you already can see what happens here. These are the GGACT Kamers. That's the M6A motif. Now, if you rank that, you can really see that among all those candidates, it's the 2 M6A writers that clearly stand out, that have the strongest impact on RNA modifications. Now we can go even further than that because we get the transcriptome-wide map as well. From this experiment, we can then identify where M6A occurs. We can see that here for metal 3 and metal 14, exactly at the 3' end. We can look into individual positions as well. And we can see here in red, that's a decrease. So we can also infer the directionality. We know whether these are writer or eraser enzymes. We can see that this is very consistent for both metal 14 and metal 3. They lead to a reduction in M6A modifications. And so really from this experiment, we can identify those M6A writers in an unbiased way, and that means direct RNA sequencing is a very powerful tool towards studying these modifying enzymes. We couple that with XPOR, which means we don't need any knowledge about RNA modifying enzymes before, so we can identify them in an unbiased way. And here we discover metal 3 and metal 14. They definitely have the strongest impact on RNA modifications. Now, one of the big questions, of course, is what about all the other candidates that do not show such a significant impact? There's, of course, some limitations, and we discussed this a lot. There might be compensation happening. It's likely to be very context-specific. We're using a HEC293 T cell line here. It could be related to knockout efficiency. Any RNA modification targeting enzyme that works on non-polyadenylated RNA, those things we would also not see. If there's no strong K-map preference, or if they very specifically target only very few RNAs, these are all things that we might not discover. And so it really shows that on one side, direct RNA sequencing is very powerful. We can really identify these enzymes with an unbiased approach here. It also shows that the epitranscriptome seems to be relatively stable, but it also highlights that there's still a lot we can learn about analyzing this large-scale data. Okay, so for the second part, I want to talk a bit more about RNA sequencing, and we have heard quite a bit about it. And one of the reasons is that there's so much we can do by profiling RNA. Just want to show a few examples here. This is a study from 2006, which is famous now because it described for the first time the induction of pluripotency. And here they used RNA profiling to demonstrate the change in cell state. A while back here, this study from 2015 is part of the TCGA studies, where they used large-scale RNA profiling and associating that with diseases such as cancer. A bit more recent here, this study from GTEx used RNA profiling to associate that with genetic variation. And so this really highlights the breadth of questions that we can ask with RNA profiling. And one of the reasons it's so popular is because it's a relatively simple assay. We need an RNA sample, and then we can do library preparation and sequencing, and all of that is commercially available. And that means it's really widely available and cost efficient. And we can use that to measure the number of RNA molecules in a sample. And really that means that RNA sequencing is one of the most widely used genomics assays. Now, to me, RNA is really interesting because it's not as simple as what I show here, but each gene can generate different RNAs. And that's because of alternative start sites, alternative end sites, and also because of alternative splicing. And so the question is, is that actually relevant? Okay. I want to show that with this example here. This is a study we did a while back where we looked into alternative promoters. And I think it's a nice example that really illustrates why we should think about transcript expression. This is the HER2 gene, or ERBB2. Here I'm showing two different promoters. And you can see, depending on which promoter is used, different RNAs will be generated. And so we profiled RNA expression driven by alternative promoters for more than 18,000 RNA sequencing samples, and then we associate that with patient survival in cancer patients. We look into the first promoter here. We do not see much of an association. When we look into the second promoter, we do see a very clear, significant association with patient survival. Now, there's two things we need to learn from this example. The first is that two RNAs, even though they're transcribed from the same gene, they can clearly be independently regulated. And they can also be functionally different. Now, why do we still talk about gene expression? And so I want to introduce transcript and gene expression, and that also explains why gene expression can be helpful. So here we have one gene with two transcripts, and in a nice scenario, our RNA sequencing data might look like that. And then we can quantify transcript expression by counting the reads that we assign to each of these transcripts. And in our case here, transcript 2 would have 4 reads. Now, gene expression, we like to think of the number of RNA molecules from all transcripts of a gene. And so gene expression here would be also 4. All of these 4 reads come from that gene. Now, RNA sequencing data doesn't always look like that, and sometimes it might look like this. And so in that case, we can't really assign those reads to any of these 2 transcripts, but we can still quantify gene expression very robustly. And that's why we often use gene expression. It's a robust way of quantifying what happens in the cell here, but it's a concept. It's not what happens, really. Okay? But it's a useful concept. And one reason I showed these 3 examples here is actually because they also show the evolution of RNA technology, starting from microarray, and then short-read sequencing towards long-read sequencing. And so for the remaining part of this talk, I really want to talk about how long-read sequencing has changed the way we analyze RNA profiling data. I want to introduce this project here, which was just published earlier this year, the Singapore Nanopore Expression Project. We wanted to generate a resource, a very comprehensive resource for long-read sequencing data and also short-read sequencing data. And so we profiled these 7 cell lines, each with multiple replicates, using different RNA protocols, including direct RNA sequencing, amplification-free sequencing, PCR sequencing, and also short-read sequencing. For most of these samples, we actually use the same RNA, which we then split and sequence with different protocols so that we can compare them. And the first thing that I want to talk about is a bit about the comparison of long-read and short-read sequencing. How much of a step is that really towards profiling transcript expression? And I want to start by looking into gene expression. Okay, that's the easier part. We just want to know how much RNA is transcribed from this region. And we use spike in RNAs where we know what's expected. That's what you can see here on the x-axis. On the y-axis is what we estimate from long-read sequencing, the 2 left plots, and then the right plot, illuminate that's short-read sequencing. But one thing that you can see is it's actually pretty comparable. There's maybe a bit more noise for short-read, but overall, for gene expression, this looks really comparable. Now, how does this look if we look into RNA from human cell line? And again, the correlation is very high between long-read and short-read sequencing. So it actually, again, looks very comparable for gene expression. Okay, so now let's look into this task, which is a bit more difficult. How does it look if you want to quantify transcript expression? Again, we start here with spike in RNAs, where we know the ground truth. But you can see between long-read and short-read sequencing, maybe again a bit more noise for the Illumina data, but overall it looks good. And so that means for spike ends, it seems that they're actually comparable. Now, how does this look if we look into human cell lines? And now we do see a huge difference. So short-read and long-read really largely disagree on transcript expression estimates. And so only one of these 2 technologies can be correct. We asked us, I want to find out a bit more about that. And for that, I want to introduce this concept here of the major isoform. So the major isoform, that's the most highly expressed transcript for each gene. Now, we do not know which isoform that is, but we get an estimate. one from long reads and one from short reads. And if they agree, we do see a very high correlation as expected. But the difference really comes from when they disagree. Those genes where long read sequencing predicts one isoform, that's what you can see here, and short read predicts another isoform as the major isoform. And so one hypothesis, of course, is that this might be related to read fragmentation. And so that's what we wanted to find out with the following experiment here. We used the long read sequencing data, we aligned it to the transcriptome, and then we did some error correction and fragmented to shorter read lengths to really simulate short read sequencing data. And then we compared that fragmented long read against the long read. We see how this looks. So as a reference, this is how it looks when you compare long read to short read data. And this is how it looks if you compare the long read data to the long read data that was fragmented to simulate short read data. And you can really see it looks very similar. And that really indicates that fragmentation contributes to the difference between short read and long read. Now this shows it just for one example, but we did this for all the samples in this project. And so here that shows the mean relative difference. Ideally, this should be 0 because we just used the long read data. But when we fragment those long reads, what we see is that it's the short read isoforms, the major isoforms that were only identified by short read, that suddenly are estimated to be expressed, even though originally they were never supported by long reads. And so that really indicates that fragmentation contributes to the difference in long read and short read transcript abundance estimates. But there's one more thing that we did here, and that's that we wanted to validate those with digital PCR and qPCR. This shows one example. In red, that's the major isoform identified by long read. In blue is the major isoform identified by short read. We designed primers for the unique component and then performed digital PCR here. And I want to show the results with this plot. On the x-axis, that's the concentration that we measured for the major isoform identified by long read. So that would be like a point for long read sequencing. On the y-axis, that's the concentration measured for the short read isoform. Okay, so any point here in the upper triangle, that would be a point for short read sequencing. And so this is how the data looks when you validate that. And it's very clear that digital PCR confirms largely what we observe with long read sequencing data. If you look into the correlation, it's also very high between sequencing and digital PCR. Whereas for short read, we do see that in the case of a disagreement, digital PCR largely confirms what we observe with the long read sequencing data. And so we do observe large differences between short and long read sequencing data. We think long read tends to more robustly estimate the major isoforms, whereas short read might show some overestimation due to fragmentation, and validation really confirms that long read seems to be more accurate. Now, there's definitely cases where short read might be more accurate than long read as well, but overall it really confirms that long read is really a very powerful tool towards profiling transcript expression. Okay, so this project actually includes a lot of other things as well, and you can look into splicing within cell lines or across cell lines. There's a huge amount of isoform diversity. We also briefly looked into fusions, and I briefly mentioned that later again. And we can use that data to look into RNA modification as well across these cell lines. I also want to mention this pipeline here. I'm a big fan of the NF-Core project, and so we contributed to this NanoSeq pipeline. And anyone of you who's interested, we really look for people who are happy to contribute to those pipelines as well. Now, one of the ideas with that project was that we wanted to generate a resource that's useful for the community, and wanted to share that very early on. About 5 years ago, maybe we started to release some of the first data sets. At that time, actually, using my Dropbox account, which I thought was a great idea. And until people used the data and Dropbox started to block my account and decided it was not a good idea. And so I'm really thankful for AWS for jumping in. And I think it's much better that it's now shared on AWS to the Open Data sponsorship program. It's about 40 terabytes of data, and you can very quickly access that. And I have my Dropbox account back, so it's also good. We also want to keep that resource alive. That means we update it with more recent data sets, including some of the single cell data sets that we plan to upload there as well, that I want to mention a bit later. And also new sequencing kits. Just showing a result here from one of the beta tests that we currently do for a new version for the Oxonanopore PCR cDNA kit, where you do see a substantial increase in read length. So you can sign up, and we'll post updates once we make those data sets available. I know there's a few of you who develop methods, and we host tutorials on that webpage as well. And thanks a lot for those people who already contributed some tutorials. And For those of you who are interested, you can include your own tutorial as well. It might help you to advertise your tools, and we're very happy to host those. You just need to submit a pull request. So there's just one more slide here that I want to show from that project. And there's this figure, which I think is really important to understand long-read sequencing data. And it shows the number of transcripts that all these reads are aligned to. And so in pink, that's short-read data. And you can already see why short-read data is more challenging for transcript quantification. That's because a lot of these reads are assigned to a larger number of transcripts. The best that we want to see is, of course, this number one. These are all the reads that are assigned exactly to one transcript. These are the unique reads, and that's where long read really has an advantage. But one reason I show that plot also because I get asked a lot about this part here. How comes that there is like such a high fraction of reads in the long read sequencing data that we can't assign to any of those isoforms? And some of the ideas and suggestions were about maybe alignment error or noisy reads, but that's not the case. We can just cut those reads short and suddenly we can assign all of those reads to isoforms. That's really a property of the read length. And so to illustrate that, I'd like to show this example here. These are two genes that are transcribed in opposite direction. And this is how the RNA sequencing data looks for that example. And you can see for the first gene, it's largely one isoform that's expressed here, maybe with a bit of noise, but not too difficult to identify which isoform that is. Now for the second gene here, and it's the same data set, you can see there's much more variation in the splicing patterns. And a lot of those reads we can't assign to any of these annotated isoforms. And that's really a property of long-read sequencing that's substantially different from what we see with short-read sequencing. And so one of the important questions is really, which of those reads represent valid RNAs, and should we add them to the annotations? And this brings us, of course, to the topic of transcript discovery. And so we developed this method that was mentioned, Bamboo, that defines this as a machine learning problem. So we represent those reads with a number of features such as read count, relative read count. We look at the standard deviation for the start or end site, also at other features related to internal poly-A priming. And then essentially we calculate a probability for each read that it should be added to the annotations based on what we observed based on annotated transcripts. And so we get this continuous probability score, and we can then use that to extend annotations. And it allows us to add anything that's missing, and that really helps to assign those reads to the right isoforms and avoid some errors. So to illustrate that, we did the following experiment here. We used one RNA sequencing data set, and we quantified it twice, once with a complete set of annotations and once with missing annotations. Okay, other than that, it's the same data set, and you can see what happens here. In gray, those are the annotations that we removed. Okay, so they're estimated to be 0, they're just non-existent. Yeah. But there's one more thing that you can see here in blue. It's the same data set, the same annotation, but the quantification is still different. That's because reads get wrongly assigned when there's missing annotations. Now with transcript discovery, we can partially rescue that. So if we turn that on, we can see what happens. First of all, some of these gray points get rescued, so you can see them in green, so we can now quantify them. We also introduce some false positives. Okay, that's expected, that's here in purple. But one more thing that you can see is that these blue points now get closer to the diagonal, which means we add those missing annotations, and that avoids reads being wrongly assigned. We can increase that threshold, and you can even see more of these isoforms being rescued. You can see that those blue points get even closer to the diagonal, but of course you get more false positives as well. But that's really one of the differences between long read and short read. For long read RNA sequencing, we always recommend to do transcript discovery. First of all, you can identify these novel transcripts, and there might be a lot of interesting things that you can learn from those. But even if you're not interested, you can essentially ignore those green points here, and you can ignore those purple points here, but you still get higher accuracy in your quantification because you better assign those reads to transcripts. There's one more effect where this is important. I've introduced gene expression as somewhat the sum of transcript expression. Now, if we estimate transcript expression and we don't assign some of these reads, they are not counted for gene expression. So this is based on spike in RNA, is what we expect and what we observe if we just sum up transcript expression. So what about all those reads that we don't assign? In Bamboo, we have a function that calculates gene expression that considers all of those reads so that we really measure gene expression based on that region. You can see including those really improves gene expression estimates. Bamboo also provides what we call full-length read or full splice match reads, unique reads, and total read counts, and that's something that we find very helpful with long-read data. You know, if you have a CPM count, that's one thing, but if you look into the data and you know how many full-length reads you have, that actually provides a lot of information. So that's something that we find very helpful as well. Yeah, we compared Bamboo. I don't want to talk too much about it. First of all, this plot is already but older, and some of these tools here, including Bamboo, have been substantially updated. But one thing that we see is with this continuous threshold, we get this dynamic range to really say we want to do very precise transcript discovery, or we want to be more sensitive. And that's something that's really nice with this approach. We get the continuous probability for each of these transcripts. Okay, so the last part here I want to talk about is what happens if we want to apply that to highly multiplexed data such as single cell or spatial data. There's two challenges. The first is the high dimensionality. We have more than 200,000 transcripts and thousands of cells. You know, that was just mentioned, and it's an engineering challenge, and it's a very important one. These tools have to be fast enough. But I don't want to talk about that too much today. The sparsity of the data is another challenge, and I think that's interesting because that really is fundamentally different, again, between long read and short read data. Going back to that example, imagine you have these reads, okay? You can see that it's largely coming probably from, in that case, transcript 1. There's a few reads that we can't really assign, but the EM works, the expectation maximization, works by looking at these unique reads and then making a decision about these ambiguous reads where to assign them. Now, if you have sparse data, your data might actually only look like this. And now you don't have any information to properly assign that read, and you might just assign it to both of these isoforms equally. And so it's really that lack of data that's challenging for the EM algorithm. Now, we might not want to have these estimates for each single cell. We might be fine by just summing it up at a pseudobulk level. And the question is, does that really rescue this? And so we did the following experiment. We used bulk data, so we have a reference, and we used that to simulate single cell data sets based on these 4 cell lines. And then we estimated expression for each single cell, calculated that based on a different number of cells within each cluster, and compared the correlation with what we observe in bulk. And you can see the larger the number of cells, the higher the correlation. However, it saturates at some point, and it's not as high as we want it to be. And so it's clear that it doesn't rescue everything. And so the way we address that in Bamboo is by keeping these barcodes. Okay, so we track all of those reads. We first perform transcript discovery across all of those reads. We assign those reads to the transcripts, and then we demultiplex them. But at this point, we only count gene counts and transcript unique counts. This is something that we can't really do wrong. Now with those, we can do cell clustering, and only after the clustering, we perform the EM. Okay, and now we can assign those ambiguous reads, and that's very powerful because now we have much more information. We have enough unique reads to make more informed decisions. And if we compare that to performing the EM on single cells, we can really see that this increases the correlation with what we expect. So that's what we call the clustered EM, and it really helps in increasing the accuracy. The more data we have, the better the EM estimate. So this is the last part that I want to mention because I think it's really a very nice illustration of how long-read RNA sequencing can be used. And it's going back to gene fusion genes. So they occur in several cancer types, and they're a consequence of genetic rearrangements. They can be oncogenic, and here I'm showing the Philadelphia chromosome that leads to this BCR-ABL1 fusion transcript and chronic myeloid leukemia that leads to uncontrolled cell growth. And from long-read sequencing, we can identify those fusion transcripts, and we can do that with single-cell RNA sequencing data. And so here we sequence these different cell lines for which we know where this fusion occurs, but we can identify them purely from single-cell long-read RNA sequencing data. If we look into one of those as an example, you can see really, we can identify it very specifically to, in that case, the MCF7 cell line. And so I think that's really a nice example to show how these long reads are very helpful. We can identify both gene expression, we can identify transcript expression, including these alternative splicing patterns, but we can also identify these genetic arrangements and the consequences that they have on the transcriptome. Okay, so that's the really last part I wanted to talk about. The summary here, I've talked a bit about a comparison of long-read and short-read sequencing, where I think that long-read sequencing really provides more robust estimates for major isoforms. Some of the differences between long-read and short-read is that we generally recommend transcript discovery. You might want to ignore it, but still, I think it's good to do it. It really is an opportunity to generate these context-specific annotations. And I've talked a bit about Bamboo, which provides this continuous transcript discovery parameter, which also estimates full-length counts, unique counts, which might be very helpful for you. It has a particular way of estimating gene expression, which I also think is important to consider for long-read sequencing data. Then I've talked a bit more about the processing of single-cell data or other highly multiplexed samples. And yeah, with that, I'd like to thank everyone involved here, the Bamboo team, who really spends a lot of time on trying to address questions and maintaining it and improving it. And all my other team members, that's Chening, André Sim, Ploy Patawanich, Christopher Hendra, UK, Min Hao Ling, Sui Yu, as well as a large number of collaborators. And yeah, I'm very happy to take questions.
Hello. Hi. Thank you for this great work, presenting this wonderful work. I have questions probably more technical. So the long read really opens like a new world in terms when you're looking at the sequencing. For example, we have seen so many like internal intentions, and also when you look into the 5' and 3' locations, it's really just a distribution. So when it comes to in terms of identifying new isoforms, how do you deal with those situations?
Yeah, I think that's a very good question. I mean, you identify, the more you sequence, the more you identify. And I think some of the speakers have shown that very few of these isoforms that we identify are very highly expressed. Those I think are clear cut, they might be very interesting. A lot are very lowly expressed, and then that's the question of how relevant are those. Then we do see a lot of variation that's, we don't even know are these two different transcripts in terms of alternative start sites or alternative end sites, and those things also get more difficult to identify. But I do think that's an opportunity to really identify all of these aspects that were challenging, and I think that's an opportunity to look into those. But yeah, I do think it's a question really of how much of those is relevant, which ones of those are relevant. But yeah, there's a lot we can identify. We've talked a lot about human data, mouse data, which is very well annotated. One thing where I think this is very powerful is actually sequencing new species, such as plant species or other species where the transcriptome is not very well annotated. And here I think this is already can directly apply. It's very powerful to identify these transcripts with high accuracy.
So I wonder if you have any strategies to deal with the situations I mentioned already, or you think that's to be ongoing kind of?
Yeah, no, in Bamboo, the way we deal with it, we just calculate for each transcript candidate, we calculate like a probability score. And I think that's important. So essentially, you can tune that score yourself. You can just decide it, similar to other genomics tools, corresponds a bit to like the idea of an FDR, depending on how many false positives do you want to include in the data set. So you can really include more or less. It also depends in the end on the question and what one wants to do with these data sets. But yeah, I think it's really important to have this kind of scores to understand how likely are these, how important are these. But of course, it's difficult to say whether these are artifacts or biological noise. And so, I think this is something that's more difficult to say.
Yeah, thank you.
Thank you. And number one.
Hi, Jonathan. Thank you. Amazing talk. My name is Satrio. I'm working in adaptive sampling. As you mentioned earlier, that direct RNA use large input, and the throughput is much lower than the cDNA version. So my question is, what is your opinion on adaptive sampling in modification study or in transcriptomics profiling in general? Thank you.
You know, I think this is, I think it's a really, really cool feature to specifically target RNAs or DNA of interest. We haven't used it ourselves, and I think one of the challenges with adaptive sampling is that it reduces the throughput, and so there's a bit of a trade-off in terms of what you target and how much you lose. Yeah, but I do think it's very powerful, and it helps it to increase throughput and look at specific regions. It really depends on the trade-off of how much do you lose versus the enrichment. Yeah, but I think it's a really cool feature. I think it has a lot of potential.
Thank you.
Oh, there's one in number 4. Okay, number 4, please.
Hi, thanks for the talk. It was great. Just question on the, for example, on the disagreement between long and short reads on the major isoform. Is there a plan to maybe look at it from a proteomics based evidence? And if not, maybe why not?
Yeah, no, we don't. I mean, I think one limitation is in terms of technologies. It's easy to profile RNA. Yeah, we don't have so much experience with the proteomics side. I think it could be helpful, but I think we can already confirm the RNA sequence. So in terms of those differences, I think that's probably sufficient. But proteomics would also help in terms of studying these novel isoforms that we identify. So I think there's a huge potential in terms of understanding what's the what can be translated. Sometimes I also wonder what exactly we measure. Is it how much RNA corresponds to protein? I think there's a lot of questions about those as well. Yeah, I think proteomics technologies are a bit more challenging, maybe. So I'm not an expert on that. But yeah, I think having these high quality proteome maps could be very helpful to associate that with RNA in general, yeah.
Thank you.
Okay, I'm gonna go for with an online question, okay, because we should not forget the online people. So could you estimate the minimum number of reads for a cell line sample like Elizabeth Sands did for the PacBio, like how many reads we need to do a single cell? Long reads is 5 million.
For single cell specifically?
Yeah. How much are you using?
Yeah,
okay.
So we use one flow cell for an experiment. So we get, I think, probably like 20,000 reads. It really depends on the question or on the money, I guess. So the more the better. Yeah, we, I guess, have in the range of 20,000 reads for each cell. Depends on the diversity also. So if you don't have too many cell clusters, you can pool. If there's a huge diversity of cells, you might need more. But of course, also is the question of do you want to sequence more cells or more information per cell? So it really depends on the question.
Okay, thank you. So we can go to number 2 now.
Okay, thanks for the talk. I'm so interested in the RNA modification. So I have 2 questions. First is about the IVT datasets you used to train your model. May I ask the complexity of the IVT datasets? Like how many transcripts in your training datasets?
Yeah, it's not very complex. It's based on short sequences that are in the range of 100 to 150 bases. They're designed to cover all k-mers. I don't remember exactly the number, but it's not something that's difficult to generate. So it covers all the k-mers, but it's not too difficult. So we get, yeah, it's a relatively simple data set. And that's what one of the ideas that we had. We wanted to see that we can generate something that's relatively easy to generate. Yeah, we have to cover all k-mers, but it works already, with relatively simple libraries.
What do you mean by all the k-mers? You mean all the 5-mer or all the 9-mer?
Yeah, we got asked that question before as well. It doesn't matter so much. Here it's based on 5-mers. The latest base calling models use, I think, 9-mers, but it doesn't really make a difference to us. There's much less information in the 9-mer compared to the 5 or 6-mers. Yeah, so yeah, we don't need to cover all of those. Yeah.
Oh, okay. I have another question about, is it possible to distinguish that the only one modification in the 5-mer or by modification in the 5-mer or the triple modification in the same 5-mer?
You ask about the same modification or different modification?
Maybe different or the same, or never mind.
Yeah.
Maybe there's a 2 M6A in the same 5-mer or 1 M6A and M5C in the same 5-mer. Is this possible to distinguish this?
Yeah, so we don't have that data, right? So we have to decide between 100% modified and 0% modified, because if you do this, it's very easy to generate. Once you want to have very specific modifications, like these kind of mixtures, that's much harder. And so that's why we don't have really data to answer that. I would think it should be possible. We do predictions anyway per base, which means we can identify one base, and then we move to the next base, which is for us another k-mer, which we can identify. We can see that we can identify these modifications for each k-mer. So in principle, I think it should have the same accuracy. We think of it as per base, not per k-mer in the end.
I see. Thank you. Thank you.
Okay, I'm sorry. I think that we are now over time, so I think that we should stop here. We can thank all the speakers of the sessions and all the speakers of today at HiTSeq.